Good day, everyone. Today, we are going to discuss an important topic in medicinal biochemistry, which is the cell and its biochemical organization. This topic forms the foundation for understanding how the human body functions at a molecular level, and more importantly, how drugs interact within the body. Let us begin with the basic concept of the cell. The cell is the smallest structural and functional unit of life. Every process that keeps us alive, whether it is energy production, protein synthesis, or response to drugs, occurs at the cellular level. In the human body, we are made up of eukaryotic cells, which are complex and contain a true nucleus along with specialized organelles. Each of these organelles has a specific biochemical role, and together they create a highly organized system. Now, before we go into the structure, we need to understand what the cell is made of. Chemically, the cell consists of both organic and inorganic components. The major inorganic component is water, which makes up about 70 to 80% of the cell. Water acts as a solvent and provides the medium in which all the biochemical reactions takes place. It also helps maintain the shape and temperature of the cell. Along with water, electrolytes such as sodium, potassium, calcium, and chloride are also present. These are very important for maintaining osmotic balance, nerve conduction, and enzyme activity. Coming to the organic components, these includes proteins, lipids, carbohydrates, and nucleic acids. Proteins are extremely important because they serve both structural and functional roles. Many enzymes, receptors, and transporters are proteins. Lipids are mainly involved in forming the cell membrane and also acts as energy stores and signaling molecules. Carbohydrates provide energy, especially in the form of glucose, and also contribute to the cell recognition through glycoproteins. Nucleic acids, which includes DNA and RNA, are responsible for storing and expressing genetic information. Now let us move on to the structure of the cell and its organelles. The first structure we will discuss is the cell membrane. The cell membrane is a selectively permeable membrane that separates the internal environment of the cell from the external environment. It is mainly composed of phospholipid bilayer where the hydrophilic head face outward and the hydrophobic tail face inward. Embedded within this bilayer are proteins that perform various functions such as transport and signaling. This arrangement is often described as the fluid mosaic model. The cell membrane plays a crucial role in maintaining homeostasis by controlling what enters and leaves the cell. And it also contains receptors that interact with various substances, including drugs. Transport across the cell membrane occurs through different mechanisms. Passive transport does not require energy and includes simple diffusion and facilitated diffusion, where substances move along the concentration gradient. Active transport, on the other hand, requires energy in the form of ATP and moves substances against the concentration gradient. There are also bulk transport mechanisms such as endocytosis, where substances are taken into the cell, and exocytosis, where substances are expelled. Next, let us talk about the nucleus. The nucleus is often referred to as the control center of the cell. It is surrounded by a nuclear membrane and contains chromatin, which is made up of DNA and proteins. Inside the nucleus, we also have the nucleolus, which is involved in ribosome synthesis. The nucleus stores genetic information and controls all cellular activities. It is also the site where transcription occurs, meaning DNA is converted into RNA. Moving on to the mitochondria, these are known as the powerhouse of the cell. Mitochondria are responsible for producing ATP, which is the energy currency of the cell. They have a double membrane, and the inner membrane forms folds called cristae, which increases the surface area for energy production. Interestingly, mitochondria have their own DNA, which makes them unique among organelles. They also play a role in programmed cell death known as apoptosis. Next is the endoplasmic reticulum or ER. There are two types of endoplasmic reticulum, rough endoplasmic reticulum and smooth endoplasmic reticulum. The rough endoplasmic reticulum has ribosomes attached to its surface and is mainly involved in protein synthesis. The smooth endoplasmic reticulum does not have ribosomes and is involved in lipid synthesis and detoxification processes, especially in liver cells. The Golgi apparatus is another important organelle. Its main function is to modify, package, and transport proteins and lipids. After proteins are synthesized in the endoplasmic reticulum, they are sent to the Golgi apparatus, where they undergo further processing and are then packaged into vesicles for the transport to their final destination. Lysosomes are organelles that contain digestive enzymes. They are responsible for breaking down waste materials, damaged organelles, and foreign substances. This process is essential for maintaining cellular health and is involved in a process called autophagy, where the cell recycles its own components. Peroxisomes are similar to lysosomes but are mainly involved in detoxification reactions. They help break down hydrogen peroxide and are involved in the metabolism of fatty acids. The cytoplasm is the fluid matrix that surrounds all these organelles. It is the site of many metabolic reactions, including glycolysis. Within the cytoplasm, we also have the cytoskeleton, which is a network of protein fibers that helps maintain the shape of the cell, supports intracellular transport, and plays a role in cell division. Now, when we talk about the biochemical organization of the cell, we are referring to how all of these components work together in a highly coordinated manner. One of the key features of this organization is compartmentalization. Each organelle performs specific functions which prevent interference between different biochemical reactions. For example, energy production occurs in the mitochondria while protein synthesis occurs in the rough endoplasmic reticulum. Another important aspect is enzyme localization. Enzymes are not randomly distributed, they are located in specific parts of the cell where their substrates are available. This increases the efficiency of biochemical reactions. Metabolic pathways are also highly organized. For instance, glycolysis takes place in the cytoplasm while the Krebs cycle occurs in mitochondria. Finally, all these processes are tightly regulated. The cell uses enzymes, hormones, and feedback mechanisms to control the rate of biochemical reactions. This ensures that the cell functions efficiently and adapts to changing conditions. So, to summarize, the cell is not just a structural unit but a highly organized biochemical system. It contains various components and organelles, each with specific roles, and all of them work together to maintain life. Understanding this organization is essential for understanding both normal physiology and disease processes. Thank you.